Greetings. My name is Chaitan Khosla, and I'm a professor at Stanford University. And this is part 2 of the trilogy of my lectures on assembly line polyketide biosynthesis. In the previous lecture, I introduced you to the evolutionary biology of these remarkable assembly lines, the chemistry that happens on these assembly lines, and I gave you a general idea of what these assembly lines look like. So we looked at the 6-deoxyerythronolide B, or DEBS, synthase that is responsible for making this precursor of erythromycin. I ended the previous lecture by giving you a sense of what we think this assembly line looks like and how that insight was derived. What I'm going to start this module with is an introduction to the kinds of tools we use to interrogate the biochemistry of these remarkable assembly lines. So these assembly lines exist, as we discussed in the first lecture, in relatively esoteric sources. They usually come from bacteria whose names many of us have a hard time spelling, or sometimes even worms, or oftentimes just sequence information that was derived from DNA that was collected from some place. In order to study these systems, one of the first set of tools that we developed was to be able to take these very complex metabolic pathways, like the DEBS metabolic pathway, and put them into genetics-friendly microorganisms, hosts like E. coli. So today you can make 6-deoxyerythronolide B in E. coli by growing it in the presence of glucose as a source of energy and propionic acid as a source of all the carbon that's used to make the product, so long as the recombinant E. coli contains DNA that instructs for the biosynthesis of those 3 very large proteins that comprise DEBS. This is, for obvious reasons, a very powerful tool to interrogate DEBS, because now if I have a bacterium that makes my product for me, I can go in there for a price of a $200 kit, manipulate the DNA that encodes this assembly line, and ask what are the consequences of this assembly line. And these tools have been in our armamentarium for the better part of the past 2 decades. My previous generation of lectures talked quite a bit about these tools, so I won't spend a lot of time doing so again. But these tools have played a critical role in our understanding of the biochemistry of these assembly lines. Now, what is more challenging, but is perhaps arguably more important, is if you want to study this remarkable enzymatic assembly line, you'd like to be able to peel off the wrapper that surrounds these remarkable proteins. That is easier said than done, but today we can reconstitute the entire 6-deoxyerythronolide B synthase from purified proteins. What I show you on the lower left corner is a protein gel, an SDS page, that shows 5 proteins. The 2 proteins to the far right are the second and the third protein of the erythromycin assembly line. And each of them, as you can tell, has a monomeric molecular mass that exceeds 300 kilodaltons. The third protein, which is the first of these proteins, could not be expressed for love or money in E. coli in a form that gave adequate yields of pure protein to study biochemically. And so we had to break it up into 3 pieces, which are shown in the first 3 slides of this gel, and purify those pieces independently. And now you can put those three pieces together with the other two proteins to make a cocktail of proteins that, in the presence of appropriate substrates, propionyl coenzyme A, NADPH, and we don't use methylmalonyl coenzyme A itself. Instead, we use an in situ enzymatic generation method for methylmalonyl coenzyme A, where we use free methylmalonic acid, coenzyme A, and an enzyme called malonyl-CoA synthetase. And so when you put these 5 proteins, which have been purified, together with all these precursors in a test tube, you see 6-deoxyerythronolide B, and what I show you on the lower right is a mass spectrum of the product that has been synthesized in a biochemical equivalent of an earth, air, water, fire type of an experiment. What this allows us now to do is to probe this machine with all the power that you're used to using to study your favorite enzyme once you've purified it to homogeneity. So what I show you in this is a very simple graph that gives you a sense that we can turn over this entire assembly line in a test tube with a rate constant that's approximately about 1 per minute. So approximately once every minute, this assembly line is releasing 6-deoxyerythronolide B in a test tube that is presented with the appropriate precursors. And that is roughly the rate we might expect this assembly line to be working at inside a cell. I also want to point out, as the inset shows, this assay is remarkably efficient. Every equivalent of 6-deoxyerythronolide B has stoichiometric mapping to an equivalent of the propionyl-CoA primer that is used, and uses 6 equivalents of NADPH, whose consumption is being measured in this simple spectrophotometric assay. Okay, so we have the tools to be able to study the entire assembly line inside a recombinant E. coli-like cell. We have the ability to study the entire assembly line in a purified, reconstituted form. We also have the ability today to study the individual steps in the catalytic cycle of these modules in isolation. So recall, in the previous module, I introduced you to some of the core reactions that occur at every module. There's a reaction that we call chain translocation, where the chain moves from the acyl carrier protein in the upstream module to the module that's receiving the chain. And if we want to interrogate just that reaction for one module, what we do is we pull out that module from the rest of the assembly line, purify that to homogeneity, present it with a chemoenzymatically derived acyl carrier protein that has the growing polyketide chain substrate bound to it, and we put it into a test tube so that the chain translocation event, the movement of that growing polyketide chain into the module, is the slow kinetic step. And everything after that that leads to the turnover of this module is fast. And so you can use this kind of an assay to interrogate, using established kinetic paradigms, that chain translocation step of your favorite module that you're interested in. The same approach can also be used to kinetically isolate the chain elongation event that I introduced you to in the earlier lecture. So when we want to study chain elongation, what we do is we take the module whose elongation biochemistry we want to study, and we prepare just the keto synthase together with the acyl transferase from that module as one protein. We produce its carrier protein, its acyl carrier protein, as another protein. We put these two proteins together. we present the two substrates into this assay, and we look for chain elongation, which gives rise to the product. And we do this under conditions where the step we want to probe, the elongation step, is the slow step, and everything else is fast. You can do exactly the same thing to probe the acyl transfer, the selection of that building block, methylmalonyl coenzyme A-derived building block from metabolism. The same approach can also work over there. And all of these assays are well developed. They're in the literature, and you can use them to study your favorite assembly line. In addition to those core reactions, chain translocation, chain elongation, and acyl transfer, I mentioned there are auxiliary reactions which I lumped under chain modification. Those reactions include ketoreductase types of chemistries. In this particular assay, I'm adding the ketoreductase, or KR, as a standalone protein to the rest of my system so that I can control the rate at which that step occurs, and I can look at the consequences of putting one ketoreductase in my assay as opposed to some other ketoreductase. And that allows me to interrogate the ketoreductase reaction. You can do the same thing at the level of the dehydratase reaction, which follows after the ketoreductase reaction in certain chain modification sequences. So all of these assays are also set up. The point you need to recognize is that you can probe through, again, a divide and conquer approach, the chemistry happening at any one of these steps in the overall assembly line. Using this combination of in vivo and in vitro tools, there are a number of important problems you can study. In the remainder of this second module lecture, I will talk to you about some examples of questions, long-standing questions in the field having to do with the specificity of these assembly lines. I'll give you two examples of those problems because they have engineering implications. And then in the next lecture, we'll talk about what the assembly line mechanisms. So stereospecificity is probably one of the most fascinating features of these complex polyketide antibiotics that these assembly lines make. So to the right, you're seeing the 6-deoxyerythronolide B product of DEBS. And for those of you who are looking at that now, you're noticing that it has 10 stereocenters. That is 2 to the 10 possible chiral forms of the same chemical formula, or slightly more than 1,000 of these chiral forms. If you go to a fermentation plant that makes erythromycin, the large vat that produces erythromycin has 1 out of those 1,000+ stereochemical forms in it, the one that I'm showing you. I think most of you would recognize that that is a really impressive feat on part of nature, how it can program this assembly line to give one and only one stereochemical outcome. That is a problem that we have quite a good understanding of how that happens today. I've cited some references on the slide, and so I will summarize for you what these references teach us about how stereochemistry is controlled by the DEBS assembly line. So of those 10 stereocenters, one of them, which is this stereocenter, is generated by this ketoreductase in module 3 of the assembly line that I introduced to you as that epimerase that looks like a ketoreductase in my previous lecture. This is the enzyme that is a homologue of other ketoreductases but does no NADPH-dependent chemistry. Instead, it epimerizes the C2 carbon atom of the growing polyketide chain that is lodged in module 3 of the assembly line. Of the remaining 9 stereocenters, 8 of those stereocenters are shown in red, and they are controlled by the 3 red ketoreductases and 1 blue ketoreductase in modules 1, 2, 5, and 6, respectively. So each of these 4 ketoreductases controls 2 stereocenters apiece. For the chemically initiated, these enzymes are not just stereoselective, they're also diastereoselective. So they're setting 2 stereocenters at a time. And these enzymes we know, these 4 enzymes we know today are both necessary and sufficient for the unique labeling of those, for the unique identification of those stereocenters. The last stereocenter, which is this stereocenter, is at the 6th position, is a more complex output, and it is generated by 3 enzymes in module 4 of DEBS. There is a ketoreductase, a dehydratase, and an enol reductase that all collaborate with each other to set this 1 stereocenter. In addition to stereochemistry, there is another very important specificity that is encoded in this assembly line at each module. And that is the specificity of that corresponds to the choice of the extender unit. In my introduction, I pointed out that all of the modules of 6-deoxyerythronolide B synthase use a methylmalonyl coenzyme A extender unit. In the case of DEBS, the R group that is shown in this enzymatic scheme would be a methyl group. Other polyketide synthases can use coenzyme A thioesters that contain other functional groups in place of a methyl group over here. And all of these choices are made by the acyltransferase. These acyltransferases are relatively specific. Not only do they have high specificity, as shown in this graph, for the coenzyme A precursor, they are picking from the metabolic soup. So what you see in this graph over here is the rate, the velocity versus substrate concentration of the preferred substrate, which is methylmalonyl coenzyme A. And down here are the rates if R is 1 methyl short, so in other words, it's a hydrogen instead of a methyl, or 1 methyl longer, which is an ethyl group. And as you can tell, this acyltransferase that we're showing you data for in this slide is highly selective for a methyl group instead of 1 smaller or 1 larger. Now, in addition to being specific for its cognate substrate, this enzyme is also specific for its protein partner, which is the acyl carrier protein that is being used. And here I'm introducing you to a concept that is going to come back in a more significant way in the last of my 3 lectures, which is the importance of protein-protein interactions in the assembly line biochemistry of these systems. In this case, what you're seeing is that the acyl carrier protein is being strongly recognized by the acyltransferase, because if you give this same acyltransferase other acyl carrier proteins from other modules of DEBS or elsewhere, they work much more poorly than the natural acyl carrier protein. So in addition to recognizing the coenzyme A precursor, you also have recognition of the acyl carrier protein. Now, for those of you who are familiar with enzyme kinetics know that from data like this, you can derive mechanisms of how these enzymes work. So in this case, this acyltransferase has a ping-pong bye-bye type of a mechanism. The coenzyme A precursor first comes in. It is bound by the acyltransferase. The acyltransferase picks the methylmalonyl extender unit. Coenzyme A leaves. The carrier protein comes in, is recognized by the acyltransferase, and takes away the product, the methylmalonyl extender unit. So that ping-pong element comes into this kind of a mechanism. Now you can also ask, in addition to these gatekeeper acyltransferases that control the choice of the building block, there are many other enzymes in this assembly line that lie downstream of each choice that a module makes of its building block. To what extent do they influence the overall substrate specificity? Do they care about what the upstream module chose as its precursor for elongating the growing polyketide chain? We can ask questions like that using the assays, biochemical assays I showed you earlier on. And from those experiments, you learned something quite interesting. So you learned that the downstream steps beyond the acyl transfer step in many modules are not that discriminatory, analogous to what Henry Ford had contemplated for his assembly line. The downstream steps have low but not a significant amount of specificity. So if by whatever mechanism you can fool this acyltransferase to put a hydrogen instead of a methyl at this position on the carrier protein, the elongation enzyme that elongates the chain and puts this R group in the growing polyketide chain primarily loses about 2 to 4-fold specificity as a result of this mistake that the upstream step made. That's not much in the grand scheme of things, but what you have to remember is these assembly lines have many, many downstream steps. So one of these assembly lines, the first module over here, or the second module, has 4 or 5 modules downstream that are looking at the consequences of what that module did. And so these small effects at the level of substrate specificity can have quite significant impact on the final product. And you can see this in the context of assays like this. So here, in the spirit of engineering, what we're doing is we're taking that natural, the natural assembly line that nature uses to make 6-deoxyerythronolide B, we're taking that full assembly line, and instead of just presenting it methylmalonyl coenzyme A, we're now presenting it a 1-to-1 mixture of methylmalonyl coenzyme A and ethylmalonyl coenzyme A. And we're asking, what's going to happen? Are you going to get just 6-deoxyerythronolide B? Are you going to get something else, 1 or 2 other products? Or are you going to get a zoo of products? And the answer to that is you get some analogs that are produced competitively with the natural product, 6-DEB. But these compounds aren't immediately obvious why these should be formed and other ones shouldn't be formed. So at least one of these, for example, this peak that I show you out here, whose mass spectrum is shown over here, we know with reasonable confidence has an ethyl group that is incorporated at the C8 position instead of a methyl group. So somehow that gatekeeper transferase has enough tolerance for an ethylmalonyl extender unit, and all of the downstream enzymes on the assembly line are sufficiently tolerant that they will let that mistake slide by so you get this desired product. And if we could predict what's going to be made and what's not going to be made through an experiment like this, why then we would have a way to precisely engineer an antibiotic like erythromycin to make a molecule like this. But right now, we're just beginning to scratch the tip of the iceberg in terms of what's possible and what's not in these kinds of systems. And an experiment like this illustrates what's possible. These kinds of insights can also be used for more sophisticated engineering experiments. Analogous to the kinds of experiments you may be familiar with when you think about incorporating unnatural amino acids in proteins that are derived by ribosomal mechanisms. And so in this, there are some examples of acyltransferases that are what we call standalone acyltransferases. They operate outside the assembly line, and because they work very, very fast compared to typical assembly line acyltransferases that exist in assembly lines, you can do some interesting experiments. So in this case, we have knocked out one acyltransferase out of all the 6 acyltransferases on the 6-deoxyerythronolide B synthase. So this is like a site-directed mutant that has inactivated that acyltransferase. And we complement it in trans with that very ultra-fast acyltransferase that we get from a different assembly line in nature. And what happens is, because this acyltransferase will pick a malonyl unit instead of a methylmalonyl unit, it can transfer that malonyl unit onto this module, because this module is simply waiting for something to come its way. And this enzyme can do it pretty quickly. And now that resulting intermediate moves all the way down in the assembly line to give you a product that has one and only one change in the entire macrocycle that is made. And what you see over here is, if I have my assembly line that is present at say micromolar concentrations in my assay, at nanomolar concentrations, I can get quite respectable incorporation of malonyl coenzyme A at that single position to give me 12-desmethyl 6-deoxyerythronolide B. So this is a way you can cheat the system so long as you have a robust enzyme that can trans-complement the module that you want to cheat. So hopefully that gives you a flavor of the kinds of tools we have and the way we use these tools to study specificity. Thank you.